For those of you who are not familiar with Fertility New Zealand walks along all New Zealanders, walks alongside all people facing fertility challenges. We have a dedicated support line and email address, and we have 3 pillars in our organisation. So we are here to provide support, information and advocacy to anybody along their way on their journey at any stage. Follow us on Instagram, Facebook or LinkedIn and keep up to date with what's happening. Membership is free to join, and it also supports the valuable work that we do in New Zealand with Fertility New Zealand. Jump onto our website. We've got lots of information on our website. We've got fertility fact sheets. All of our webinars are recorded and available online as well, and lots more information to help you along your way on your journey. Tonight, I'd like to introduce you to Jeanette MacKenzie, who is a Scientific Director at Fertility Plus. Jeanette has been working in this field for over 17 years, and has worked across all aspects of clinical laboratories. Now, if you've got any questions for tonight, please put them into the chat box and Jeanette will get to your questions at the end of the presentation. So I'm going to hand over to Jeanette.
Thanks, Kate, and thank you to Fertility NZ for inviting me to give this talk tonight, which I really hope you find interesting. So I'm going to be talking to you about what is happening in the laboratory. So first of all, what is embryology? So this is the science used in assisted reproductive technologies, and it's talking about the growth of embryos in the lab and also looking after sperm and eggs in the lab. Embryologists are specially trained scientists, and in New Zealand we are all registered with the Medical Science Council of New Zealand. It's required by law for every embryologist to be registered. And we come from a variety of backgrounds, obviously starting with science, but some people come from genetics or microbiology, or nowadays a lot of people do a master's in clinical embryology. So that is who we are. Now, going back to the very, very beginning of IVF, where did it all begin? So the first IVF baby was born in England in 1978. This baby is Louise Brown, and her birthday was the 25th of July. So her birthday is coming up very soon, and she's going to be 44 years old. Just to point out that date, the 25th of July, we have adopted it as World Embryology Day. So in a couple of weeks' time, embryologists around the world are going to be celebrating World Embryology Day, and it's a day where we might have a morning tea or a cake at work, or do other events. And here in Auckland, we'll be having a embryology get-together for all the embryologists in Auckland and have a bit of a networking event in celebration of World Embryology Day. So back in England in 1978, Sir Bob Edwards and Patrick Steptoe were the scientist and the doctor involved in the birth of Louise Brown. I should also mention there was another very important person involved, and that was Jean Purdy, and she was the nurse involved, and she had a lot to do with this as well. Moving on to 1980, Australia had their first IVF baby born, and that is Candice Reed, and she was the third baby in the world to be born. Moving into 1984, New Zealand achieved their first IVF baby, and her name was Amelia Bell. Back in the 80s, success rates were averaging about 10% per cycle. The science involved was very, very basic, and none of the techniques or culture media or anything used at that time had been developed specifically for IVF like we have in the labs these days. When Louise Brown's mother was having this treatment, her doctor told her chance of success was 1 in a million. So I'm going to give you an overview of an IVF process and then what is happening in the lab at those parts of the process. So to start off with, before we get the eggs in the laboratory, the woman is going to take some hormone stimulation. And are the injections you take to stimulate lots of follicles to grow in your ovaries, and the follicles are where the eggs grow. Once you've had scans and bloods to check that the follicles are the right size to give that final trigger injection, your egg collection is planned. Now, on the day of egg collection, the embryologist will come into theatre with the doctor, or sometimes there's a hatch between the theatre and the laboratory that the eggs can be passed through. So as the doctor's draining the fluid from the follicles, the embryologist will be taking those test tubes and looking at the fluid under a microscope to check for the presence of the eggs. On the same morning when the egg collection is happening, the partner will be producing a sperm sample. And once the sperm sample is received in the laboratory, that's when we wash the sperm. Now, we can use a fresh sperm sample, or you might have had a sample frozen, so we would thaw it out, or you might be using donor sperm, so we would thaw that out. The process of washing sperm involves us putting the sperm through a series of gradients, and I describe it like a sieve. So it starts off at the top, and then it gets spun down in a centrifuge, and all the good motile sperm will get spun right to the bottom of the tube, and then all the immotile and abnormal sperm will be stuck in those sieves along the way. So what we have at the end is a highly concentrated sample, which is nice and clean, of lots and lots of sperm. We then inseminate the sperm and eggs together, which I'm going to go into more detail about that in the next slide, and then we culture the embryos, and then we do an embryo transfer and cryopreservation and freezing. Okay, so this slide here, you can see is a cartoon overview of IVF. So we've got there day 0, which is your day of your egg collection. We've got eggs and sperm. In an IVF cycle, we would inseminate the sperm and the eggs together after lunch, usually 4 to 6 hours after your egg collection. We inseminate thousands of sperm into the dish with the eggs to allow the sperm to swim to the eggs and fertilize those eggs. So as I said, we do that after lunch, and we just put the sperm into the dish, and then the dish goes in the incubator, and we leave it alone for the night. And then the very next morning, which we call day 1 in the laboratory, the first job an embryologist does in the morning is a fertilization check of the eggs that have been inseminated the night before. So on day 1, we do the fertilization check, and what we're looking at this stage is the egg, which is one cell at this stage. And if it's normally fertilized, we see 2 circles in the egg. Those 2 circles represent the chromosomes from the sperm and the chromosomes from the egg. And if we see those 2 circles, which are called pronuclei, then we know it's normally fertilized. And you might hear us refer to a fertilized egg as a 2PN or a 2 pronuclear embryo. And that's what we're talking about at that stage. Now, sometimes we see things that are not 2PN. We might see a 1PN, which is when there might be asynchronous development of those nuclei. And it might mean that it is fertilized normally, and the nuclei have developed at separate times, or it might mean that it's trying to fertilize, but it hasn't actually fertilized. What we usually do with the 1PNs is we keep them in culture and see what they do. And if they form a nice embryo, then we know there's a good chance that it probably was normally fertilized and it's going to have a good potential for pregnancy. If the embryo, while we're watching it along the week and it doesn't really do anything, then we know it probably wasn't really fertilized. Another thing we see at fertilization check is a 3PN. So that's when we see the 3 of those circles in the egg. Now, with IVF, this generally tells us that probably 2 sperm got themselves inside the egg instead of just 1. Now, a 3PN, or one with more than 3PN, we can see 4 PNs or 5 PNs sometimes. They are abnormal and we discard those straight away. We're unable to use those because eggs with more than 1 sperm inside are incompatible with life. So the next day, day 2 in the laboratory, when we look at an embryo, we want to see something around about a 4-cell stage. Sometimes we might see 2 cells or 3 cells, 4 cells or 5 cells, but 4 cells is on average what we see. And then by the 3rd day, we're seeing something around about the 8-cell stage. Now, sometimes you might be told it's 7 cells or 9 cells, and that's okay. Not every one of the cells will divide at exactly the same time. So sometimes we might catch it at the 7-cell stage or sometimes at the 8-cell stage. And that's all normal. And then we've got day 4, which is not even written on the cartoon, because on day 4, we generally don't look at the embryos. We leave them alone in the incubator. They are working really hard to become blastocysts. A blastocyst is a day 5, 6 or 7 embryo. Now, a blastocyst is really different from the early cleavage stage embryos on day 2 and 3. By day 5, the embryo is starting to look quite different. We've got some cells which are migrating to the outside of the embryo and forming a flat layer around the embryo. These are called trophectoderm cells, and they'll go on and form tissues like the placenta and the sac. And then we have a clump of cells inside the embryo called the inner cell mass. Now, the inner cell mass will go on and form the fetus itself. And then we also have a fluid-filled cavity, and that's on day 5, 6, or 7, the blastocyst stage. Now, embryo transfer can happen either on day 3 or day 5, and your embryologist will talk to you about which is best in your situation to have your embryo transfer. And it'll depend on how the embryos are developing, and it'll also depend on what policies and protocols are in place in the laboratory where you're having treatment, as every lab has a slightly different policy about when to do the transfer and when to do blastocyst or when to do day 3. At transfer, you'll most likely be recommended to have 1 embryo transferred. In New Zealand, it's very, very common to have just a single embryo transfer back, and more unusual to have 2 embryos back. So the embryologist will select the best embryo for transfer based on the morphology or the appearance of the embryo. And then if there's any extra embryos that have also developed onto a good quality, they can be frozen. So freezing usually happens at the blastocyst stage, and freezing can take place either on day 5, day 6, and sometimes day 7. So your embryologist will keep in touch with you about how many of the embryos have been able to be frozen. And on average, we expect about 30 to 50% of fertilized eggs to become blastocysts. So as you can see, it's quite a big drop-off from fertilized egg to blastocyst. Right, so the next slide, this is describing ICSI. So you might have heard about ICSI. Now that's an acronym for intracytoplasmic sperm injection. And this is a different method of putting the sperm and egg together. An IVF cycle with ICSI is exactly the same for every part of the process except the part where we in the laboratory put the sperm and egg together. So egg collection's the same, sperm collection's the same, but the difference will be that we will be picking up a single sperm in a little needle and injecting it straight into the egg. Now, before we can actually do the injection, we need to denude or strip the egg. Now, this is because when the egg comes out of the ovary at egg collection, it's covered in hundreds of other cells that are called cumulus cells. And they're called cumulus cells because it's a big fluffy ball of cells. It looks like a fluffy cumulus cloud. Now that's fine to keep the cells on there for IVF, and we don't take them off for IVF. We put them all in the dish together because those cumulus cells are there for a reason, and they help the egg's development and help with fertilization in a standard insemination. However, when we're doing ICSI, we need to see the egg, and we need to see if the egg is mature, and we actually need to be able to hold on to it tight. So we need to get rid of those cells so we can do all those things. And that is called stripping or denuding the egg. So you might hear your embryologist talk about denuding and stripping. That's what that is. Now, when we have stripped the egg, we can see the egg itself, and we can see if it's mature. A mature egg has a little blob on the side of it, and that blob is called a polar body. And that is telling us that it's a mature egg and it's ready to receive a sperm. If there's no little blob, it means the egg is not ready to receive a sperm, and we won't inject those eggs. So on average, about 80% of eggs will be mature on the day of egg collection. So if you had 10 eggs collected, on average, we'd expect 8 of them to be mature and suitable for ICSI, and you might have 2 that were not mature, so they would be discarded at that point. So the sperm injection is also done 4 to 6 hours after the egg collection, so usually after lunch. And then the eggs are put away in the incubator for the night, just as we did with the IVF eggs. And then again, the next morning, day 1, the embryologist will come in, first job in the lab, check the eggs for fertilization, and then everything else is exactly the same as the standard IVF cycle. So that's all the very, very basic things. What other things do we do in the laboratory? So you might have heard of PGTA, and this is pre-implantation genetic testing for aneuploidy. And what this is saying is it's checking the blastocyst for abnormalities in the number of chromosomes. So every embryo should have 23 pairs of chromosomes, which is the makeup of normal cells. But sometimes things can go wrong, and there might be an extra chromosome, or there might be a chromosome missing. And this means that those embryos are going to have less chance of being a baby and more chance of having a miscarriage. So what do we do? If you're going to do a PGTA cycle, we need to grow all of the embryos to the blastocyst stage, and then we assess them for the suitability of doing a biopsy on day 5, 6, and 7. We make a little hole in the zona pellucida, which is the shell around the embryo. On the picture, you can see that layer around the embryo. So we make a hole in that so we can get inside the embryo. And then on the side of the picture, you see that it's a pipette coming in, and we remove about 5 cells from the embryo. Now, the cells that are removed are trophectoderm cells, which, remember I said before, were the outer layer of cells. And I mentioned the inner cell mass before, that was going to become the fetus. So when we do a biopsy, we stay far away from the inner cell mass. We don't take those cells. We don't want to disrupt them. So the embryologist takes about 5 cells from the embryo. These cells are put into a little tube, and then the tube is sent off to a genetics testing lab. In the meantime, your embryo stays in the laboratory, in the IVF laboratory, and it gets frozen, and then it gets given a unique identifier so we know exactly which embryo of your batch it is. Now, the cells are sent away to the laboratory, they do the testing, and that can take up to 4 weeks. So that is why we need to freeze the embryos to wait for the results to come through from the laboratory. So, is this going to increase your chance of having a baby? Well, it's not going to increase your chance of having a live birth per egg collection. But what it might do is, if you've got a lot of embryos, it might quicken the time to pregnancy. So what I mean is, say if you've got 5 embryos and we're not doing genetic testing and we're choosing the embryos based on their morphology, we're going to pick the best looking one and transfer that. And then next month, if it didn't work, we'll transfer number 2, and then we'll transfer number 3, and so on. But it might be number 4 or 5 that actually had the correct number of chromosomes. If we've got the PGTA results and we know actually embryo number 4 or 5 has got the normal number of chromosomes, we might make our decision differently and we'll transfer that embryo first. Hence, we might be able to get to pregnancy quicker. Now, there are some things to note with this type of testing, and one thing is that this test is not 100%. It's not really classed as a diagnostic test. It's more of a screening tool to help us select the best embryo. So that means that if you do get pregnant, we still recommend to have all your routine screening that you have, you know, your 12-week testing and everything when you're pregnant. It is an invasive procedure. So you can see, you know, we're physically doing something to the embryo in the laboratory. We're making a hole in the zona and we're removing some of the cells. And we have done this over and over and over again, and we get pregnancies and we get normal babies. But we just need to keep an eye on the literature and the research, and especially into the outcome of the babies, because we're not sure yet if it is causing any changes to growth in the womb yet. So the other thing is, when we do this testing, we get given the results, and there's sort of 4 possibilities of what you're going to get in your results. So the first one is it's going to come back, the result's going to say it's a normal embryo and it's suitable for transfer. Great, that's easy. Second option is I might say it's an abnormal embryo and it's not recommended for transfer. Okay, that's pretty black and white. But then there's a third and a fourth result that could come through. So the third one is called no result, and that means that we don't know the answer. We don't know what the chromosomes are, because the cells that got put in the tube and sent to the genetics lab, for whatever reason, the DNA in that tube maybe wasn't of good quality, and the genetics lab haven't been able to get an answer, haven't been able to see what the chromosomes are doing. And that happens about 5% of the time. So in that case, you've done the biopsy, we've frozen the embryos, we've done these extra procedures, but we actually don't know the answer for that embryo's chromosomes. And then the fourth result that you could get is a mosaic embryo. Now, mosaic means that some of the cells in the embryo are normal and some of the cells are abnormal. So in that biopsy, if we took 5 cells, some might show normal, some are abnormal, and it will be presented in a result of a percentage of mosaicism. So what would happen usually is we would consider those embryos for transfer after you've spoken to someone like a genetic counsellor who can discuss what those results mean and what the chances are. Another thing to just mention along the lines of mosaicism, when we've taken the sample of the embryo, we have taken 5 cells from the embryo, but those 5 cells might not represent what the whole embryo's chromosomal makeup is. And this is also mosaic. So it might be that the cells that we took for biopsy are saying that it's abnormal, but actually the rest of the embryo is normal. So you've got an incorrect diagnosis, and it can also happen the other way around. So the cells we take might say it's normal, but actually that's only a tiny proportion of the embryo and the rest of the embryo is abnormal. Hence why we can't say that the test is 100%. So in New Zealand, this testing is not publicly funded. So if you are doing publicly funded IVF treatment and you want to do this, you will need to pay an extra fee to the clinic, usually at least over $2,000. And the more embryos you have to be biopsied, the more expensive it's going to be. And I just point out that what I'm talking about here is the PGT testing for aneuploidy for chromosomes. So there is other testing, pre-implantation genetic testing for hereditary diseases. And yes, there's public funding for that, but that's not looking at the chromosomes and chance of implantation. That's looking at specific genes, and that's a slightly different process in the genetics laboratory. Okay. So what else can we do in the laboratory that you might have heard of? So I'm just going to talk about a couple of advanced sperm selection techniques, and you might have heard of this, IMSI. So that's intracytoplasmic morphologically selected sperm injection, IMSI. So that's selecting a sperm to do ICSI at very high power based on what the sperm looks like. So this is an additional tool that embryologists can use specifically for selecting which sperm is going to be injected for the ICSI process. So the ICSI process, we see sperm using pretty high magnification, which is 200 times, and that's the picture of the sperm on the right. But IMSI uses a very, very high magnification at 600 times, and that's the picture of the sperm on the left. And you can see the sperm head in a lot more detail. And what the embryologist is looking at is all the details in the sperm head, which is really an important part of the sperm because that's where all the DNA is. Are there any risks to this process? Well, nothing different to a normal ICSI cycle. Doing IMSI might be useful if you've had previous failed ICSI attempts, so it might be something different to try. Unfortunately, though, the research is not supporting the use of IMSI routinely in infertile men. It hasn't shown any overall benefits. And because this is an extra selection tool, it's an add-on, so there'll usually be an extra cost and an extra consent form to do if you use IMSI. So another one you might have heard of is PICSI, and that's physiological intracytoplasmic sperm injection. So this one is using a different type of ICSI dish, and it's got hyaluronic acid in the dish, and what we as embryologists are looking for is that the sperm are bound to these drops of hyaluronic acid and the heads bind to them, and then the ones that are bound are the ones that we want to select to do ICSI. Now, why is this important? Well, we have known in the literature that experiments have shown that the sperm do bind to the hyaluronic acid, and these sperm are more likely to have better morphology and motility, but more importantly, lower rates of DNA fragmentation. What are the risks for doing this process? Same as a normal ICSI cycle. Now, what about the evidence? What's that saying? So there was a recent large randomized study showing that PICSI didn't increase the chance of having a baby, but they did say it might reduce the chance of miscarriage. And again, this is an extra process in the laboratory. It's an add-on, so there would usually be an extra cost involved and an extra consenting process. All right, next slide. Okay, let's move on. Come on, slide, move over. Kate, I can't seem to move slides. So I was going to talk about embryo glue next, and that is another thing that you've probably...
Oh, Um, to reappear on screen. So sorry about that, Jeanette. Not sure what happened there. Can you
Okay.
see the embryo glue slide there?
Yep, I'm at the glue slide now. Shall I carry on?
Fantastic. That's wonderful. Thank you.
Okay, great. So embryo glue, that's the next thing I was going to talk about. So you've probably heard of this. It's a special medium that has got very high concentrations of hyaluronan and recombinant human albumin. Now, those are two things that we know are involved in promoting implantation of the embryo. So the glue is the culture media that we put the embryo into to do the embryo transfer. What are the risks of this? Well, no known risks for using embryo glue. And what is the evidence? Well, we do have some evidence for this one, and we know that the Cochrane review reported an increased pregnancy rate and live birth rate by about 10%. So what is the cost for this? Most clinics include this in the cost of your cycle and use it routinely for every embryo transfer. And I'm pretty sure every clinic in New Zealand transfers every embryo in embryo glue. Okay, another one you might have heard of is also a different type of culture media. It's called Embryogen and Blastgen. It is a commercially produced media. It's an alternative to the standard IVF media, and it contains GM-CSF. Now that is a growth factor, and it's specifically involved in cell division, cell growth, differentiation, and suppression of cell death. What is the evidence for using this? Well, there is some low quality evidence that it is no more or less effective than normal culture media that doesn't have GM-CSF. This is an add-on. So if you wanted to try it for something different, it will be a different cost. It'll be a cost, and there'll be a consenting process as it's a add-on procedure. Right, time-lapse imaging. So time-lapse incubators have been an amazing development in IVF labs, and embryologists love them. So they are incubators with cameras built inside them. So the camera takes a photo every sort of 5 to 10 minutes of the embryo's development, which means at the end of the day or the 5 days of embryo culture, the embryologist can push a button and watch a little video of the embryo's development. So what this means is it's really good for picking up unusual embryo development that we wouldn't normally see with just looking at the embryo once a day. And by seeing embryos with unusual development, we are able to identify embryos with a lower potential than the other embryos. Good thing about the time-lapse incubators is they're very high quality incubators. They've got very good temperature control and pH control and really good monitoring and alarm systems. They're really user friendly, and they've got the videos. And a really nice thing is patients can have a video of the embryo developing. So that's a really, really cool thing. Another thing that they have are algorithms to enhance embryo selection. Now, the studies have shown that, unfortunately, using a time-lapse incubator is not going to increase the chance of having a baby from one egg collection, but it's still a nice thing to have in a laboratory. As I said, embryologists love them. They create efficiency in the lab. And again, you can have a beautiful video of your embryo developing. So time-lapse incubators sometimes will incur an additional cost, and sometimes it might be included in your cycle costs. IVM, so this is called in vitro maturation, and this is an alternative to standard IVF. So you would have reduced hormonal injections, and the egg collection would be done before the eggs are mature, so before you have a trigger injection. Now, some women might be suitable for this if you're at extreme risk of OHSS, or it might be a cancer patient who wants to do fertility preservation and preserve her eggs, but sometimes for some cancers, the fertility drugs could make the cancer worse. So as I said, the eggs are collected when they're immature, and then they're matured in the lab. So that's an extra couple of days in the laboratory before the eggs actually are mature. For this process, ICSI has to be used for the fertilization. Now, success rates are lower than standard IVF, and so therefore this treatment isn't done very often. So there's less information available on the safety. But even though success rates are lower, it might be suitable for specific people. And if your doctor recommends it, then that might be your best option to try to reduce OHSS or get mature eggs. Okay. And then for the future, so what's happening in research at the moment? There's a lot of work going on for non-invasive PGTA. So this means trying to work out what the chromosomal makeup of an embryo is without doing that invasive biopsy of the embryo. So in research labs, they're looking at the media that the embryo is sitting in, because the embryos are some cells are going into the media, and they can test the media to see what the chromosomes are of the embryos. So it's still in relatively early stages. And there was one pilot study that showed 84% concordance with an actual biopsy. In many of the studies, the efficiency of the technique has been restricted by technical difficulties. But right now there is a very large multi-centered randomized study underway. So we keep our eye on those studies. So that was my overview of what happens in the laboratory. So if you have any questions, please feel free to let me know what your questions are, and I can answer your questions. And here they come. I can see one. Is it true more IVF babies are male than female? Why is this? Yes, there are some studies that show there is a bit of a skewed population of male embryos. So one thought between why that might be is because we think that male embryos develop slightly faster than female embryos. So what this means is if we're on day 5 and we're in the laboratory and we're picking which we think is the best embryo, it's probably going to be the one that's more advanced. So we might be more likely to pick a male embryo over a female embryo. The ratio is not too significant. We're talking about maybe 51 to 49%. But yes, that is definitely something that there's lots of literature on. Where, how in New Zealand can you get the training? Oh, so where and how in New Zealand can you get the training and the qualifications to become an embryologist? Okay, so the qualifications to become an embryologist in New Zealand, there's no specific embryology course, but there's a lot of Bachelor of Science degrees in New Zealand universities that do focus on genetics and embryology, and that's really good. And then you keep your eye out for a trainee embryology position at one of the clinics, and then you can get some hands-on training in the laboratory. Alternatively, you can look further afield at specialised master's courses that specialise in embryology. So there's a master's in clinical embryology at Monash University in Melbourne, and that's a hands-on and theoretical master's that sets you up really nicely to become an embryologist. And they've also got one at Oxford University in England, which is also a really well-known embryology course. But a lot of it in New Zealand is having a bachelor's degree with some kind of postgrad training as well, and then landing yourself, to be lucky enough to land yourself a trainee position. Can you tell if an embryo is a male or female? No, we can't tell in the laboratory by looking at it if it's male or female. if you do PGTA testing, part of the testing of the chromosomes is looking at the sex chromosomes, but the laboratory will only report it as normal or abnormal. They will not tell the embryology team what sex the embryo is, because sex selection in New Zealand is illegal. But yeah, so no, we can't look at an embryo and see if it's male or female. Could you share more about how PGD works, please? So if you want more information about PGD, I am guessing you mean the diagnosis for hereditary diseases. So the process in the laboratory is the same. It's just a different type of genetics test if you're looking for a hereditary disorder, because they're looking for a single gene. So the genetics laboratory will do a different process there. Do you get told the grades of embryos before transfer? So some clinics will routinely tell you the grade, some may not. Some may describe your embryo as poor, good, or excellent. We do have fancy grading systems, especially for blastocysts, which get quite complicated. But yeah, so some labs will say, but if they haven't said, you're more than welcome to ask for an explanation of what the grading system is in the laboratory. What else have I got? My wife and I are currently expecting our first child through IVF. We are just wondering, if we were to have another one, how is the next embryo chosen? There were, I think, 4 that were 4AA quality. How can one be chosen and others not? So if your laboratory used a time-lapse incubator, they might have some extra information to do with the scoring. If they don't use time-lapse, there will be extra information in the scoring. So the 4AA is the quality right at the time on day 5 or 6. And then if they need to distinguish between all embryos that appear the same, we would usually go back to the history of the embryo's development in the earlier stages and see which one performed more normally. Yep, I've read that one. Okay. Can you tell if
I think you've covered all of those ones now, haven't you, Jeanette?
Yeah. Okay, great.
Has anybody else got any final questions before we do head off? We are watching on Facebook Live at the moment as well, so I'm just keeping an eye out for questions on Facebook too. Great presentation, Jeanette. Really informative. It was awesome. I loved it.
You're welcome. Did something else pop up? Is there a risk to embryos with the testing? So yeah, I was talking about that it is an invasive procedure. So we are physically removing about 5 cells of the embryo. As I said, we know that plenty of pregnancies have happened, plenty of babies have born that are all normal, but it's still an invasive procedure, and there is a slight risk of damaging the embryo. The same as when we do an ICSI process, we're poking a needle inside an egg. So there's always a little risk of damaging the embryos. A little risk, as in about 1 to 5%.
Tonight, Jeanette, I think that was our last question that came through.
Great.
So I'd like to give you a big thank you for tonight. It was a a fantastic and very informative webinar. I hope everybody else enjoyed it as much as I did. And I'd also like to thank our sponsor, Merck, who helped bring this webinar to us tonight as well. So I would like to wish everybody a good night. And a big thank you to Jeanette for being with us tonight. So thank you very much.
Thanks, Kate.
Thanks. Bye.
Bye.